All right. Well, I think it's ready to get started. Thank you guys all for tuning into our webinar. My name is Chris Meny. I am the Content Manager and Director of Website Development for Gold Biotechnology. Gold Bio is a supplier of proven, published, and affordable reagents and chemicals for research labs and small businesses all over the world. We specialize in thousands of the day-to-day reagents that you need in the lab, but at affordable rates that you can discover more. Check out our website for all of our products at goldbio.com. So our webinar today is about becoming a successful scientific principal investigator. Joining us for some expert advice is Dr. Dave Ornitz. Dr. Ornitz is an alumni endowed professor in the Department of Developmental Biology at Washington University School of Medicine in St. Louis. He earned his bachelor's degree from the University of California, Davis in 1981, and he earned his PhD and MD from the University of Washington in Seattle in 1987 and 88. He pursued a postdoctoral research in the Department of Genetics at Harvard Medical School before joining the faculty at WashU. His current research uses molecular, genetic, and biochemical approaches to study the regulation of cell growth, development, response to injury, and cancer, examining growth factors like FGFs, FGF receptors, and a variety of other interacting signaling pathways in the mouse embryo and in adult mice. His laboratory focuses on skeletal, cardiovascular, and pulmonary development, physiology, and response to injury. Dr. Ornitz has had an amazing career studying cell signaling pathways and is considered one of the world leaders in the FGF family of signaling molecules. Hey, Dr. Ornitz, thank you for joining us today.
Well, thank you, Chris, for the nice introduction. And let me share my screen now.
All right. I'm going to turn my video off, and I'll see you on the other side.
Let's see. Okay, well, thank you, everybody, for tuning in. I would like to give you some of my thoughts and wisdom that I've accumulated over my career as a graduate student, postdoc, and faculty member now at Washington University. And let me begin with this introduction. So many of you have graduated from college, and you're at a point where you want to decide what to do. And you really have lots of choices, and you can fill in the direction on the sign that is going to lead to the future of your life any way you want. And you have choices for postgraduate education, such as getting an MBA or getting a PhD. And just to contrast these two examples, an MBA can lead you to a career in business with a job in finance, consulting, marketing, or as an entrepreneur. And a PhD tends to lead to a career in science, where you can also become an entrepreneur, but you can engage in research or teaching. And if you follow the research career, that can lead to a career in academic research or a career in industry research. And today you have lots of choices. So in blue on the right is the traditional pathway toward a faculty position in academia, where you go from undergraduate training to graduate student, postdoctoral fellowship, and then a faculty research position. But along the way, there are many really good job choices and opportunities that lots of people consider. And I have colleagues that are engaged in all of these different opportunities in industry, in government work, and in nonprofits as well. And today I'm going to focus on the graduate postdoc trajectory, which tends to lead toward academic or industry research positions. So a PhD, what is it? It teaches creativity and problem solving. So in this cartoon, we have this blank spot, then a miracle occurs. And this is where we have a gap in knowledge. And this is where you fit in as a graduate student or a postdoc. Now, your job is to fill in gaps in knowledge and increase the knowledge and publish your work so the rest of the world can know about what you've learned and what you can teach them. So on the path to becoming a principal investigator or a group leader in industry, I'm going to outline 10 rules that I've found are helpful. to both develop your career and lead to a balanced lifestyle. So number one, I think the most important thing is to find a good mentor. In this cartoon, I'm looking for a mentor who can show me how to get rich without boring me with a lot of advice. This is not the type of mentor you want. You want a mentor who will help you to develop your career and your life. So you want to talk to lots of potential mentors. You want to talk to mentees of potential mentors. And you want to find out how the careers of other mentees developed. And this will help you choose a mentor or mentors that are suitable for your career development. So you want to pick a style that fits your style. So you have to know yourself and know what your style is. And it's important to remember that mentors do not make good graduate students or postdocs. Students make themselves into successful scientists. So this requires your own motivation. And a good mentor will, of course, help you, but also give you the opportunity to make your own discoveries and your own mistakes. And that's how you can learn. And making your own discoveries and mistakes will help you build self-confidence and persevere through the difficulties inherent in any project or any career path. So I just wanted to give an example of the mentors that I encountered during my career development. As an undergraduate student at UC Davis, I had the real pleasure of working in the laboratory of Erwin Sagel, and he was a very rigorous teacher. He emphasized quantitative biology, and he wrote classic textbooks on biochemical calculations and enzyme kinetics. And I also published my first paper in his laboratory. As a graduate student at the University of Washington, I had the honor of working with Richard Palmiter, and I joined his lab at a time when the transgenic mouse technology was just being developed. And this was a really exciting time in science and gave me lots of opportunities to grow. And Richard was really a special mentor in that he allowed and encouraged independent thinking and independent work. And this really helped me develop to an independent scientist. And finally, my postdoctoral mentor, Philip Leader at Harvard Medical School, was an amazing lab to be in. At the time I was in his lab, it was a large lab that was well funded, and basically everybody in the lab was allowed to operate independently and just coming to Phil Leader for advice. And Phil is known for deciphering the genetic code. He worked on the genetic basis of antibody diversity. He was one of the first people to make a transgenic mouse with an oncogene, and he had the first ever patent on an animal, and it was called the oncomouse. Number two, find a project that excites you. So as a graduate student in the 1980s, a project that I chose was to ask whether a small piece of DNA could direct cell-specific gene expression in vivo in a mouse. And to do this, I took a small piece of the rat pancreatic elastase gene promoter, and I fused it to the human growth hormone gene, which is a nice compact gene with 5 introns and exons, and made a transgenic mouse with this in collaboration with Ralph Brinster's lab at University of Pennsylvania. And we found that this transgene construct allowed expression of human growth hormone in pancreatic acinar cells, showed in bright green, and no expression in pancreatic islets, which are the dark islands in the middle, and no expression in any other cell in the mouse. So this was the first demonstration of cell-specific gene expression in a mouse. So this led to lots of questions. So how do you like my new genes? So we have transgenic mouse technology, but this technology is great, but are the genes expressed? Are they regulated properly? Are they altered during inheritance? So this was some of the key questions that we asked back in the 1980s, and I've been involved in answering these questions to various degrees ever since. Number 3, believe in your own abilities. So a quote that's adapted from Mark Twain that I like: a person cannot be comfortable without their own approval. So you have to believe in yourself. You got into graduate school because you have great abilities and potential. And this is the time to develop confidence in your abilities. Number 4, remember that 90% of success is dealing with failure. So when experiments fail, it does not mean that you have failed. But there are 3 things that will increase your chance of success: persistence, persistence, and of course, persistence. So if you're not making mistakes, then you're not doing anything. I'm positive that a doer makes mistakes. A quote from John Wooden. And a quote from Albert Einstein: if we knew what it was we were doing, it would not be called research, would it? So as a graduate student, I had a couple of questions that I wanted to ask. And one was, can we direct gene expression to the liver? So we made transgenic mice with an albumin promoter driving human growth hormone, similar to what I showed you with the elastase promoter. And this was before PCR, so we used a technique called solution hybridization to detect mRNA levels. And what you see on the right side is a lot of low numbers near background. So this is a failed experiment. This piece of promoter and DNA didn't work. And I did thousands of these assays trying to look for something that did work. And I should say eventually it did work once we used a big enough piece of DNA. But in contrast, when we used the elastase promoter and I plugged in different tissues from mice, we see where I bracketed the pancreas that we have nice big numbers compared to the low background numbers. And this was the first experiment that I did in graduate school that was successful. And when I saw this result come off of the counter, I knew that I would eventually graduate from graduate school and get my PhD. So this was a really happy day. Number 5, impose structure on your day and your life. So you're responsible for your own success. It's up to you to get up, go to work, and get things done. They won't happen on their own. Set daily, weekly, and monthly goals. And I like to actually write notes down on a piece of paper, for what I'm going to do tomorrow, what I'm going to do next week, and then on my computer calendar, what I'm going to do in the next month and the next year. Number 6, take ownership of your project, physically and intellectually. So in this cartoon, I expect you all to be independent, innovative, critical thinkers who will do exactly as I say. So this is not the way to learn. This is not the way to take ownership of your project. You've got to be responsible. You can't depend on somebody telling you what to do. So you're the driver of your education. You're no longer in the passenger seat. Your ultimate goal is to figure out on your own what you need to do and why, do it, interpret the results, and do the next step, and then go and tell your PI why this is the best way to go. So this is a process that takes time to develop. And when you achieve it, you are ready to become independent. Number 7, talk to lots of people, build networks. So some of my best scientific colleagues I met during graduate school and as a postdoc. And ask lots of questions of everyone and yourself. Take the time to sit under a tree and think about your experiments. What do they tell you? What do they not tell you? What should I do differently next time? Sitting under a tree or lying down is very therapeutic. Gives you a time to really think about what you've done and what you're going to do next. Number 8, read a lot. So even old stuff from 20 or 50 years ago, or even older, most ideas have been thought of before, but now we have the technology to tackle them. And I don't know who came up with this quote, but I like it a lot. Remember, a week in the library could save you a year at the bench. So it's important to read and to know what's out there. And a quote from Louis Pasteur, chance favors the prepared mind. So as an example, in the 50s, Waddington published this very famous image of the epigenetic landscape, where he proposed that a cell, as it courses through development, makes decisions as it rolls down this landscape. And this is a pathway to make differentiated tissues. And he proposed that underlying this epigenetic landscape are a bunch of genes, which are these little black pegs at the bottom. And these genes are basically tugging at the landscape to sculpt the course of development. And this is what he could do and think about at the time. But now with modern genetic tools, we can actually probe the function of cells as they progress through the epigenetic landscape. We can look at all the genes. We can actually redirect cells to go in both directions and reprogram and differentiate under different trajectories. So with early concepts developed many years ago, we can now apply modern technology to really understand at the molecular level how cells behave in an organism during development and during regeneration. Number 9, work hard. So graduate school is a privilege. It's not a right, and you need to earn the PhD. Take your work seriously. Don't take shortcuts. Remember, there's never enough time to do something right the first time, but there's always enough time to do it over. A quote from John Bergman. And do lots of experiments, but do thoughtful experiments so that everyone counts. And lastly, number 10, have fun. Enjoy what you're doing. Take some time to go fishing or whatever. It'll give you a new perspective. Take care of yourself. Listen to your mother's advice. Pursue a hobby, exercise, eat well, etc. And remember that your goal might seem a long way in the distance, and it might seem like it will take forever to get there. But if you proceed toward that goal one step at a time, eventually you'll get there. And then you can look back and see where you came from, and you'll realize that you've come a long way and that you now have a bright future ahead of you. So thank you very much. I appreciate you joining this webinar, and I'm happy to take any questions that you might have. And I'd also like to thank my colleagues at Washington University and around the world for their mentorship, collaboration, and inspiration that contributed to this presentation. And I want to thank all the graduate students and postdocs and lab staff that I've worked with over the years for all their dedication, hard work, insight, and enthusiasm that makes running a laboratory such a rewarding experience. Thanks, and we can take some questions now.
Thanks, Dr. Ornitz. So one question is, as you're going through the process of looking for a mentor or just finding a product that you want to, there's a lot of networking that goes involved in that. Do you have any advice for how students should try to network? Who should they go to? Like, what should they be asking? How do they get around that?
Well, if you're just starting out in graduate school, most graduate programs really try and get you involved and talking to and interacting with other graduate students that are more senior to you, as well as your colleagues. And talking with other students, you can really get an advice on the faculty in your graduate program and get an idea of who are good mentors to choose from. And then, it's really good to go and talk to faculty and learn about their research and, learn about their mentoring style, talk to people in their laboratory and see how successful they are. And then, eventually choose a lab. And then the other thing that occurs in most graduate programs are the opportunity to do rotations in different laboratories where you spend several weeks to a couple of months working in a laboratory to really get a feel for how that laboratory operates and how the people in that laboratory operate. And this is another good way to help choose a lab and help choose a mentor.
Well,
and then once you're in a laboratory, we have mentoring committees that, in addition to your primary mentor, that help guide you through your graduate program. And at the postdoctoral level, we have postdoctoral mentoring committees as well.
So how would you? How do you decide whether how do you decide whether you're going to stick with academia or whether you're going to just go ahead and bite the bullet and go to industry?
Well, first of all, I want to say that both options are really good options. And I have colleagues that are in academia, of course, and I have many colleagues that are in industry, and people who've graduated from my lab have gone in both directions. So, one thing is to think about what you want to do and how you want to approach science. So in academia, you have a little bit more freedom to choose your direction and change directions and do what you want. You have a life where you have to pursue funding, so this is stressful at times. And in industry, you have the opportunity in many companies to pursue basic science research as well as more practical research. The topic of your research is a little bit more constrained by the nature of the company. You're not applying for grants in industry, but you have to justify your work, and so this can be stressful at times. And these are both good options, and they're worth exploring, and they can be explored at the postdoctoral level in both academic and industry settings.
Right. And we just did a webinar a couple of weeks ago on Dr. Eric Samuels, who did a nice presentation on how to switch, how to make that jump from academia to industry. So if you guys are interested in looking at that, I would look up our webinars. We have those on our website, and you can find all of our webinars there. So here's a good question, and this is one that I've had myself. So how do you overcome the anxiety of embarking on something that's just so new and so innovative that there's really no precedent for it?
Well, this is what's really exciting about research. You can come up with an idea, and hopefully you're in a position in your laboratory, in your university, where you have a little bit of resources to pursue wild ideas. There are also some funding mechanisms, both through the government and private, that give some funding to do completely off-the-wall exploratory research. This is really one of the great opportunities, and it can be pursued both in academia and in some cases in industry. You're given the opportunity to find, what's called high risk, high impact research studies.
Right. Well, then, just as on the flip side of that, then how do you deal with when your research just kind of drags on? Like you were saying, in your research, you went through a lot of small molecules with negative results after negative results. How do you deal with that amount of failure? Like you feel like you're in a rut, like the research isn't going anywhere. How do you know whether you need to cut ties with it or change or pivot off, or how do you deal with those kind of situations?
Well, back to my slide: persistence, and persistence. This is really important, and I use it every day. It's good not to put all your eggs in one basket. So, even as a graduate student and postdoc, have a primary project, but you want to have some backup projects, and it could be that your primary project is a little bit more risky.
Right.
And then you might use some backup projects, which are a little bit, more of a sure thing. They may not be quite as exciting, but they'll get you data, they'll get you publications, and they'll help you move to the next step. So we all have to take risk, and we have to temper that risk with, some projects that will offer some more security.
Right. Now, I know you and I have talked about mentors previously on a couple of occasions, and so just to bring back to this conversation. So I know there's lots of horror stories of having a bad mentor. Having a mentor who just seems at odds with you all the time. So it's like, and I know you went into talking. It's like you've got to talk to their mentees, and you've got to really kind of look around and figure out what that mentor is like. But what happens if you try to do all those things and you still fell in with a mentor that just isn't supportive at all? They just seem like they're frustrating you at every level, and they don't really seem like they care so much about your experiments. So how do you deal with that?
Well, unfortunately, what you're saying is true, that sometimes we don't make the best choice for whatever reason, and we end up in a laboratory with a mentor that is just not a good fit. And we have a couple of choices at this point. First of all, as a graduate student, and to a lesser extent, but also as a postdoc, we have mentoring committees. So groups of other professors in the university who meet with you a couple of times a year and go over your projects. They're people that you can go and talk to. They can be your secondary mentors, and you can get advice on how to deal with the situation. If you really find the situation is untenable and you're not happy or not making progress, my advice is to bail out and switch laboratories as soon as possible. And hopefully you've learned from this experience, and you can find a different laboratory that is a better fit with your style and your goals, and find a mentor who is going to really help you succeed, which is really what's important.
Awesome. So one says, so as a graduate student, is it make it or break it to obtain your own rewards and grants, or is there some kind of grace that you get being a graduate student in that situation?
So a graduate student time is, and a postdoc time is really a wonderful time in your career, because you're under much less pressure to fund your research. We encourage all of our graduate students to apply for graduate fellowships, and there are ones through government, through the NIH, and through private foundations. And this is great practice to learn how to write grants and proposals, and to get criticism back, and to experience failure, and to experience success. At the postdoctoral level, we encourage the same, where there are postdoctoral fellowships to apply to, and small grants. And also at both stages, you can work with your mentor to help your mentor write grants, both government and non-government grants. And this is not directly your proposal, but you can have a lot of input and a lot of learning experience in helping to write proposals. So the important thing is, during this period of time, your training period of time, is to do a lot of work in the lab and enjoy what you're doing, learn lots of techniques, and at the same time learn a little bit about what you're going to have to do in the next steps of your career.
So as far as time commitment goes, what is a general time commitment that you think it takes? We know how long it takes to get through grad school. What's the time commitment after grad school, after you get your PhD? Do you think that it takes you to become a principal investigator?
Well, in academics, we hopefully enjoy what we're doing, and we do put a lot of time and effort into thinking about our projects and doing our projects. So, I'm thinking about my projects in my laboratory 7 days a week, and sometimes I dream about projects. So it's really part of your life. But hopefully, it's something that you enjoy doing, so it's not always a burden. There are stressful times. Writing grants, waiting for grants to become reviewed is certainly stressful. But then I should also stress that it's important to take a vacation now and again, and I certainly do that. And it's good to get away. And I exercise regularly, and this really helps clear my mind for thinking about things. And so, keeping a balance in your life is important, even though, and you can do this as well as thinking about your research and your work all the time. You can clear your mind by doing other things also.
Right. All right. Well, I appreciate it. I think that's all the questions we had for the day. Thank you so much for taking your time out to do this webinar for us. I hope all the attendees got a lot out of it. And if you guys have any questions, feel free to reach out to Dr. Ornitz or to us, and we'll get you in touch with Dr. Ornitz if you need any help or advice.
Great. Well, thanks, Chris. It's been a pleasure being on the air today. And again, I'm happy to talk to people and provide advice and mentorship whenever possible.
All right. Thank you guys all for watching, and we are signing off. Thanks again.
Bye-bye.